Okay, this slide has to do with prokaryote growth and reproduction, which occurs quite a bit differently than it does in eukaryotes. So this is one bacillus cell. It's still one cell. It's in the process of cell division, however. And when cell division results in reproduction, we refer to it as binary fission. Binary meaning two, fission meaning split. So simply means split in two. And it's quite a bit different than eukaryotic cell division that involves mitosis. If you've ever studied mitosis, it is a process of nuclear division before the cell actually divides. Prokaryotes have no nucleus, therefore there is no mitosis going on here, so don't make the mistake of referring to this as mitosis. Mitosis only occurs in eukaryotes that have a nucleus, because mitosis is nuclear division. So again, this is binary fission, and it's a form of asexual reproduction. No two parents are involved here. No sperm and egg are involved here. No fusion of two cells to produce a new individual. All of those things define sexual reproduction. So during prokaryotic binary fission, the parent cell elongates about twice its size. So that's when growth occurs in prokaryotic cells. Growth occurs shortly before the cell undergoes binary fission. At the same time, DNA replicates. So we go from having one circular chromosome. If you'll recall, bacterial cells have one circular chromosome. That replicates, and we get two circular chromosomes, and each chromosome then is going to migrate to each of the two new cells. And we call these two new cells daughter cells. At this point, this has not completed binary fission, so this is still considered the parent cell. But it's going to divide into two new daughter cells, and the way that happens is that new plasma membrane and new cell wall forms between the two cells, and they eventually separate. The two daughter cells that result from this are genetically identical. Replication of that one circular chromosome means that both daughter cells are going to get an exact copy of that circular chromosome. The parent cell no longer exists. Hopefully that's obvious to you, that if we split this parent cell into two daughter cells, then the parent cell is no longer going to exist, which is quite a bit different than in sexual reproduction. The parents still exist. The daughter cells will not grow. Again, the parent cell doubles in size, and then it splits, and the daughter cells are the size they're supposed to be. So there's very little, if any, growth at all after after binary fission is complete. The advantage of this type of reproduction, like any asexual reproduction, is that it's very efficient. There's no requirement for two parents, no reproductive behavior, no energy goes into producing sperm and eggs, and many more offspring can be produced in a much shorter time than in sexual reproduction. So there's a huge advantage to it. There's also a pretty big disadvantage, and that's reduced genetic variation or diversity, which, as you know, is the raw material of natural selection and evolution. However, this binary fission happens so often, and bacteria reproduce so often, as in as little as 20 minutes. They can undergo binary fission, and as you can imagine, this can increase their population or increase a colony of bacteria very quickly. All of that DNA replication going on can result in mistakes, mutations, and that's one source for the variation that we see in prokaryotes is mutation. So because they reproduce so often, there's a greater chance in a shorter period of time of mutation, and that will introduce variation and diversity into a colony or a population's gene pool, as we say, and that then will be acted on by natural selection. So my point is that bacteria, even though they reproduce by binary fission, which is an asexual process, they still can evolve very quickly. And that's a problem for us, because they can evolve to make us sick, and they can continue to evolve to make us sick. So there are also other forms or other ways that prokaryotes can increase their genetic variation and diversity, and that's through transferring genes between individuals. And we refer to that as horizontal transfer. And there are 3 different kinds of horizontal transfer. The primary one is known as conjugation. So this is prokaryotic genetic recombination, and we refer to sexual, the gene shuffling that goes along with sexual reproduction. We refer to that as genetic recombination too. So both prokaryotes and eukaryotes have forms of genetic recombination. In prokaryotes, it's primarily or solely by horizontal transfer, what we refer to as horizontal transfer, and that means between individuals. The transfer of genes between individuals. No new individuals are being created. It's not reproduction. It's just the transfer of genes. So the process that we see taking place in this transmission electron micrograph is conjugation. There's a pili bridge that is formed between these 2 individuals, and genes are being transferred from the individual on the left to the individual on the right. It's not a form of reproduction. No new individuals. The cell on the left has more pili. As you can see, all these little hair-like things sticking out are known as, the pili and fimbriae are kind of synonymous terms. So it's a form of genetic recombination, horizontal transfer, across a pili bridge. And if you recall, when we were labeling the structure and function of prokaryotic cells, when we labeled the pili, that's one of the functions that we listed for pili is the process of conjugation. And now you're looking at it. And what usually is transferred across the pili bridge is a copy of the plasmid. So again, when we were looking at prokaryotic structure and function, when we labeled the plasmid, we made a note that it was involved in conjugation. The information that's being transferred will include the gene for forming fimbriae or pili. So notice that the cell on the right doesn't have a lot of pili or fimbriae. So one of the capabilities that's being transferred through this conjugation is the ability to form pili and fimbriae and go on and conjugate with other cells itself. So the ability to conjugate is one of the things that's being transferred here. Unfortunately for us, what also can be transferred from one prokaryotic individual to another is antibiotic resistance. In other words, if this cell is resistant to antibiotics, it can pass that capability on to other cells in its colony, and that can just be passed and passed and passed from individual to individual until the whole colony is antibiotic resistant. And if that is if those are the bacteria that are infecting your body, then it's going to be really, really hard to get rid of that infection. So during conjugation, genes are transferred, but it's not like the transfer of genes is going to cause these 2 cells to become genetically unique, or genetically identical. They started out genetically unique, and only some genes are being transferred from one individual to another, so they're going to remain genetically unique. It's not like binary fission, which results in cells that are genetically identical. So another form of horizontal transfer is something that we learned about previously. Remember Griffith's transformation experiment, which was involved in the discovery that DNA is the genetic material? Transformation is simply the taking in of DNA from the surrounding environment. And I don't have a figure here for that. But if there's DNA in the surrounding environment of a bacterial cell, the bacteria can actually take that DNA in. So if a bacterial cell, a prokaryotic cell, dies and lyses, breaks open, spilling its DNA into the environment, other bacterial cells, other individuals can actually take in that DNA. And that's transformation. Another one is transduction. Now, here we go with these trans words again. You know, it's like translation, transcription and translation when we're dealing with the expression of genes. Here we have transformation and transduction. It'd be difficult to keep these straight. So transformation is when DNA is taken in from outside the cell. Transduction is kind of like the accidental transfer of genes by bacteriophages. So this figure is showing what happens in transduction, where a bacteriophage will infect a cell, and as you know, new phages will be produced within that cell, and they'll accidentally pick up pieces of the host DNA and transfer that to the next cell. So the next cell will be transduced and get genes from the previous cell. So this is carrying genes from another bacterium. So a cell was infected before this cell, and this is a virus that's coming from that cell that was infected, and it's accidentally carrying genes from that cell, and it's going to then introduce those genes into this bacterium and transform it, transduce it into a cell that now carries the genes from that previous bacterium. So the advantage of horizontal transfer for prokaryotes is genetic variation and diversity. It allows natural selection and evolution to act on them. And as I mentioned, they actually evolve very quickly. And for humans, it's genetic engineering. We can use these processes to get genes into bacteria that we want to get into the bacteria. For example, the gene for insulin, the human gene for insulin, can be inserted into a bacteria, and that bacteria then will express that gene. Even though it's a human gene, the prokaryotic cell has the machinery, has the capability of expressing that gene and making the protein, making the insulin for us. And that's just one example of how genetic engineering has been used to make products for us, for humans. The disadvantage for humans is the transfer of genes for antibiotic resistance, like the scenario that I suggested here in conjugation. That's a bad thing for us. Good thing for them, bad thing for us. So, of course, bacteria are everywhere. Well, how do they get everywhere, and how do they survive everywhere? Well, part of it is because they can form these things called endospores. So this is an endospore. This is a transmission electron micrograph showing a bacterium forming an endospore inside. And notice that the outside appears to be breaking down. And that's what happens. The endospore forms, and then the rest of the cell outside or around the endospore just breaks down, leaving behind just this little endospore. And it is even smaller. I mean, prokaryotic cells are already small, but the endospores are even smaller, and they can easily be transferred or carried on wind and by water and moved from one place to another. Here, these are colorized transmission. This is a colorized transmission electron micrograph, and so that's the endospore forming in this cell. This is the endospore forming in this cell, and again, you can see that the cell around it is lysing. It's breaking down, and that will leave the endospore behind. So what's in that endospore is simply DNA and a little bit of cytoplasm, and then it's all surrounded by cell wall material and plasma membrane. So plasma membrane and cell wall material surrounds the endospore. They form when environmental conditions become unfavorable. So say, for example, a bacterium is living in a moist habitat, and if that moisture dries up, the habitat dries up, then the bacterium might form an endospore to survive that unfavorable condition. The rest of the cell around the endospore lyses or breaks down, and the endospore can remain dormant for a long time, even millions of years. They have found bacteria that have been trapped deep underground or in ice that they've been able to revive after hundreds of thousands or even millions of years. So they can remain metabolically inactive for a long, long time until conditions become favorable again. That's what dormancy is. Dormant just means metabolically inactive. So they're not using energy. They don't need to take in any materials or generate any kind of energy to stay alive because they're metabolically inactive. they eventually germinate. So we refer to spores germinating just like we refer to seeds germinating. And germination is simply the process of leaving metabolic inactivity and becoming metabolically active again. So when environmental conditions become favorable, then they germinate and start to undergo binary fission, and one endospore can give rise to a whole colony of bacteria. So the primary purpose of endospores are to survive unfavorable conditions. But the secondary purpose is to disperse. And again, this is the same term we use for seeds. Spores disperse, seeds disperse, and it simply means moving sometimes great distances. There are bacterial spores that make their way up in the atmosphere, and they can travel all over the world on the winds and in the atmosphere, and they'll eventually settle back down and possibly find favorable conditions to germinate. So that's one reason why bacteria are everywhere.